Let's talk about bacterial gene expression. Now, note here we're back on the prokaryotic side, so we had just finished a little section on eukaryotic transcription and how much more complex it is. But gene regulation is no different. Luckily, you're here with the prokaryotes, and we don't have to deal with the eukaryotic gene regulation too much. So, all of the gene expression studies, the initial studies were all done on prokaryotes because it's so much easier. Now let's think about what gene expression is, or the regulation of gene expression. Why would we even need this? So a cell has a certain number of genes, and they specify proteins. Now there are some proteins that the cell needs all the time, certain enzymes that are needed all the time. So those are typically expressed at a fixed rate, or they're just housekeeping genes that the cell needs all the time. So these are constitutively turned on genes. They are always on. Then there are some genes, some protein products off of those genes that the cell only needs occasionally. So if we don't need them, why would the cell make them? It costs a lot of energy to make proteins, and so if you don't need that protein or an enzyme or whatever it is, then why would you want to make it? So cells are very efficient, and these genes, they need to be induced or repressed. And so basically, we're going to take a look at how this works. We're going to take a look at the inducible genes, repressible genes, so how these genes are being regulated. And again, luckily, we're here in the prokaryotic side, and it's relatively simple. So basically, we're going to take first a look at pre-transcriptional control. So basically, before we get a transcript, what can the cell do to only transcribe what it truly needs? Okay, and basically the model for the prokaryotes is that you have repressors and you have inducers. And so we're just going to take a look at the old Jacob-Monod model of gene regulation, which is the first model that was worked out, and from there on people started to understand more how genes are being turned on or off as needed. So in repression, we have a repressor that inhibits gene expression and basically blocks transcription of a certain gene or several genes at once. An inducer is an on switch basically that turns on the expression or the transcription of a gene. Now the difference is if you have a repressor, that the default position of that particular gene is it's in the on state unless the repressor turns that gene off. Now the initial state of an inducible gene is that inducible gene is normally off, but when the inducer is present, then you get transcription and translation of that particular gene. So there is this Operon model of gene expression. Now in prokaryotes, a lot of genes, they're clustered together depending on what the cell wants to accomplish. So usually the metabolism of certain food molecules, for example, requires more than one enzyme, and the enzymes that are necessary for one particular food molecule, they're clustered together. The initial model with which this was worked out is the lac operon. So that's all the enzymes that are necessary to metabolize lactose, the sugar lactose. Okay, so the operator is the segment of the DNA that controls transcription of structural genes, and the operon is a set of genes, basically, that accomplishing a certain metabolic pathway, for example. So let's start out with the inducible operon. In this case, the set of genes is turned off in the off switch state unless the inducer is present. And so coming back to this example with the lactose metabolism, normally let's say you have E. coli, and normally it feeds on glucose, and it doesn't need any enzymes that would be able to metabolize the lactose. So, but now let's say you're feeding the E. coli, you're putting lactose into your broth, and now E. coli says, oh, I would like to metabolize this, but now I need enzymes in order to process the lactose. And so now the lactose itself becomes an on switch, an inducer for the enzymes that are necessary to process the lactose. So in the presence of lactose, the lactose being the inducer, we're going to switch on the set of genes that we need in order to metabolize the lactose. So the lactose will bind to a repressor that has turned off this set of genes, and then the repressor will come off of the typically the regulatory region, the promoter region of that set of genes, and then we get transcription. So this is what this looks like. So here is the entire operon, where we have the structural genes Z, Y, and A, and they're controlled by the promoter and the operator right there. And then here you have a regulatory gene I that's ahead of this whole of the operon, basically. So the operon itself consists of these structural genes plus the promoter and operator that control these structural genes. Okay, so now what happens is in an inducible operon, here's the sort of the steady state. We have a repressor protein that's blocking the RNA polymerase from making a transcript of these genes Z, Y, and A. Okay, so in order to get the RNA polymerase to go past this roadblock right here, we have to remove the repressor. Now the repressor is being removed by the inducer. Okay. So the default here in these inducible operons is that they're in the off state because there's a repressor that's blocking transcription. So now here comes the lactose, the inducer. That's this stuff in green right here, and it will bind to the repressor. And the repressor now changes the shape, and it will come off of the operator right there. So take a look at up here. So here we had the repressor was in place. It was blocking transcriptional activity. It didn't allow the RNA polymerase to move down here and do a transcript of genes Z, Y, and A. So now here comes the inducer. It binds to the repressor. The repressor changes shape, and it comes off of the regulatory region here, right there, the operator. And now that the roadblock has been removed, now your RNA polymerase is moving on and making a transcript of Z, Y, and A genes. And now you have the messenger RNA here. So the transcript is done. You have the messenger RNA of these 3 different genes. And now by translation, we're going to make the beta-galactosidase, the permease, and the transacetylase, those 3 enzymes that are necessary in order to process the lactose. And this is really a very convenient kind of model for gene regulation, because you can see that the repressor only comes off when there's lactose present. As soon as the lactose is metabolized, then the repressor will be reactivated. It will block the operator right here, will move right there, and it will block the RNA polymerase from making any more transcripts. So now let's move on to repressible operons. The default state for them is that they are on, typically. And so now here their state, it looks like this. So the default is that you get transcripts of these structural genes E, D, C, B, and A, so 5 here in this case. And there's no block, no hindrance right here. The operator is open, the promoter is open, so RNA polymerase can do happily its job, and it will make these structural genes. So here we have the RNA polymerase making transcripts all the time of these genes right here, and by transcription, then translation, we're going to get the protein products out of these genes. Now note that you have this inactive repressor protein right here. So that came from this region I here, this regulatory gene. If you do transcription and translation of this regulatory gene right here, then you get the repressor protein. Now again, in these repressible operons, the default is that this one is inactive, the repressor is inactive, and we do get transcription and translation of these structural genes. Now we need to somehow activate the repressor, and here is a mechanism for that. Here we have a tryptophan amino acid, And that one is listed here as a co-repressor, and it will bind to the repressor. So when tryptophan is present, it will bind to the repressor molecule and now activate it. And so now it fits into the operator region right here, and it will block the transcription of these structural genes. So it will block it will form this roadblock for the RNA polymerase, and now we don't get the transcript anymore. So in the presence of tryptophan, so this is then a co-repressor, tryptophan will bind to the inactive repressor, activating it. And then once you have this active repressor, then it will bind here to the operator region. And finally here, let's take a look at positive regulation. So a lot of times what happens is that cells use something called catabolite repression. So in catabolite repression, a cell is inhibited from using carbon sources other than glucose, as long as glucose, of course, is available. So then you want to use the glucose if possible. Now, if glucose is not available, then some metabolite will build up. Here in this case, the example is given cyclic AMP. It builds up when the glucose is not available, and then that building up of the cyclic AMP, it will initiate the genes that are necessary for other sugars to be processed, for example, lactose. So in this case, the cyclic AMP would then bind to the lac promoter for all the enzymes that you need for the lactose processing, and then you get transcription and translation of all the enzymes that you need for the lactose metabolism. Here's an example of how this works in using E. coli. So the first panel right here shows the growth rate of E. coli just using the sugar lactose, and then here the growth rate of E. coli when you're using glucose. And so of course they grow much faster, the bacteria grow much faster with the sugar glucose. Okay, now let's say we have a culture and there's a limited amount of glucose. Now here the culture is growing, cells are dividing and growing really fast, and then they're running out of glucose, and now the metabolite will accumulate. So the cyclic AMP is accumulating right here, so you're going to have a little bit of a lag time, and before we can use an alternative sugar, the lactose that's still present, we need to first do transcription, translation. The regulation, of course, has to first happen. So now we need to remove the blocker from the lac operon, and now we can utilize, once we have the genes that the proteins, the enzymes that we need to process the lactose, then we can just keep growing again. Okay, and here would be a model showing the positive regulation of the lac operon in the way that we just talked about. So as soon as the cyclic AMP, the metabolite here, as soon as it accumulates in the cell, then we are basically using we're activating the cap here, and then we're just going to bind this to the promoter and initiating the transcription, and then afterwards the translation of those structural genes from the lac operon that are necessary to metabolize the lactose. Other ways of controlling gene expression would include epigenetics and then also post-transcriptional control. So let's take a look at epigenetics first. Epi means on top, and genetics, well, you already know. So on top of the genetics, so basically outside of the encoding of the nucleotide sequence, there's basically the influence of the environment on gene expression. And it happens mostly by adding chemical groups like a methyl group to the DNA, and that then can turn genes on or off. In the case of methylation, if you add methyl groups to DNA, then genes are being turned off, but that methylation can also be removed. And then lastly, post-transcriptional control via microRNAs. They would base pair with mRNA to make a double-stranded, and then the double-stranded RNA is enzymatically destroyed, And that is shown here in this diagram where you have a microRNA and it's binding to the mRNA right here, making it double-stranded, and then that double-stranded portion is destroyed by enzymes and degraded, and the mRNA would be degraded.